And the next category is antiviral agents interfering with cellular penetration and early replication. When we looked at these, at the cycle of the virus, we said there is attachment, there is penetration, and then of course it gets inside and it starts this synthesizing its structural proteins, its functional proteins, its structural proteins, and the nucleic acids, and then they all come together and assemble. Once they assemble, they leave. So, but before, because it cannot divide outside the host cell, it must be able to penetrate the host cell and get inside. So there must be mechanism. It has to have mechanisms for being able to penetrate the host cell. And because those mechanisms will be unique for the virus, they can again act as targets for the drugs. And that's what we are looking at. And we have drugs like amantadine, which also we have looked at in the management of Parkinsonism. There is also rimantadine, which is rimantadine, which is just a methyl, an alkyl or methyl derivative of amantadine. There is also oseltamivir and zanamivir. These are what we call neuraminidase inhibitors. They inhibit a very specific enzyme that is found in the virus, which you do require for these viruses to be able to penetrate the host cell. So we have those four main drugs, which we will look at. The first one, that is amantadine. Of course, it's the same structure that we did mention. You can see it's like you have quite a number of rings, but I would take it like I have this ring there and that ring there, and this cuts through. So you have the whole of that ring there, and you have that ring here, and you have also this ring on that side. So the amantadine is just as we had discussed in management of Parkinsonism. It's a cage-like, it's caged, but of course the mode of action here is different, as we are going to see. When you look at rimantadine, as I have said, it is a methyl derivative. One of the hydrogens here, it's a methyl. It's an alkyl derivative. It's an alkyl derivative of amantadine. Then you have zanamivir, which we have said has a different mode of action, and oseltamivir, which inhibit specific enzymes within the virus. And rimantadine is more active than amantadine. Definitely, it has a huger alkyl group. I believe the alkyl group helps it in attaining higher lipid solubility, better penetration. But we did mention some characteristics of amantadine, and we did mention because of the cage-like, the fact that it is cage-like. Although it is protonated at physiological pH, it still has very high lipid solubility. It's able to penetrate the lipid membranes effectively. It have two mechanisms in common. They inhibit an early step in viral replication, most likely viral uncoating. In some strains, they affect a later stage that probably involves viral assembly, possibly by interfering with the hemagglutinin processes. So those are the two areas they will affect: uncoating, early penetration, most probably the uncoating, and also a later stage whereby they are supposed to assemble, and this could be because of affecting this hemagglutinin. We are going to mention it. It is indeed a protein that is found within the virus. The main biochemical locus of action is either the type A virus. So these ones, the amantadine and rimantadine, are only effective against influenza type A. And they act on the virus M2 protein, which is an integral membrane protein that functions as an ion channel. So they block it. It acts as an ion channel. It allows the movement of protons, which is required for the uncoating. The main biochemical locus of action is the influenza type A virus, M2 protein, which is an integral membrane protein that functions in an ion channel. The M2 channel is a proton transport system. By interfering with the function of the M2 protein, the adamantines inhibit acid-mediated dissociation of the ribonuclear protein complex early in replication. So all we are saying is that these drugs will block the proton channels and interfere with the basic acidic environment, and of course, with interfering with the basic environment, they interfere with the formation of the complexes that are required for uncoating and, of course, early penetration of this microorganism, of these viruses. They also interfere with transmembrane proton pumping, mainly maintaining a high intracellular proton concentration relative to the extracellular concentration, enhancing acidic pH-induced conformational change in the hemagglutinin during its intracellular transport at a later stage. So they're saying the first one was interfering with the function of the M2 protein. The adamantines inhibit acid-mediated dissociation of the ribonuclear protein complex early in the replication, and also, as we have said, they are also interfering with the pH within the cells or the virus, and that pH is necessary for the intracellular transport, or it is important in the processes of this virus. The conformational changes in hemagglutinin prevent transfer of the nascent virus particles to the cell membrane for exocytosis. We are going to see, once you assemble these proteins or these viruses, once you assemble them, they must move to the cell membrane, and they must attach themselves to certain proteins, or to certain receptors within the cell membrane. And once they do so, they are able to move out. So all we are saying is that one of the very important proteins from the virus is the hemagglutinin. The hemagglutinin is very, very important. And once you have changes in these proteins within the virus, it will not bind the cell membrane of the host, and therefore allow for, we are going to see sialic acid and so on. It will not bind the cell membrane of the host and then allow for it to move out. They move out through exocytosis, of course, come in through endocytosis. So we are therefore saying these drugs are interfering with the M2 protein. They are also interfering with the hemagglutinin, and they are also influencing the pH within the virus, and therefore interfering with the changes, or the interfering with the hemagglutinin, and therefore interfering with it being able to help in the transport of these cells or to the outside of the host cell. So they have quite an advanced way. These drugs have quite an advanced way of, and like you can remember when we were looking at them at amantine, or also amantadine, when we were looking at it in terms of Parkinsonism, it was very easy. It was just getting in, allowing dopamine to get out, and therefore increasing the levels of dopamine. So here it is binding a certain receptor. It's binding a certain receptor, which we are calling the M2 protein, because proteins are just enzymes and or receptors. That receptor, and most of the receptors are also enzymes, binding it, causing conformational changes, interfering with the acid movement, and therefore interfering with the processes of the virus, both including its transport, so that, because finally it must leave the nucleus, move out, and get to the outside so that it can attack other viruses. Resistance with inhibitory concentrations have been associated with single nucleotide changes that lead to amino acid substitutions in the transmembrane domain of M2. All we are saying is that you can have, again, amino acid, almost like single nucleotide changes, that you can have one nucleotide being changed. People have been associated with single nucleotide changes that led to amino acid substitution in the transmembrane domain. So within the domain M2, you have an amino acid change. And once you have remember we are saying they are proteins, an amino acid change will not allow for this drug to bind. So you just have an amino acid chain within the receptor, and once you have an amino acid chain, then the receptor is different. So in other words, they can have change within the receptor by an amino acid chain, and once you have that change, then the drug cannot bind the receptor, and therefore the virus will have its activity even in the presence of the drug. And both amantadine and imantadine share cross susceptibility and resistance. So once that is resistant to one, don't bother to give the other. So this is just what we have said. I think perhaps this could be even a simpler way of putting it. They block the migration of protons into the interior of the virions within the endosomes. The endosomes are complex intracellular vesicle system for endocytosis. They therefore preventing the pH shift required for uncoating. It's the same thing we have said, that you require the proper pH within this virus. And once there is no proper pH within the virus, there cannot be uncoating. And once it doesn't uncoat, definitely it cannot penetrate the host. It inhibits the early stages of viral replication by blocking the uncoating of the viral genome and the transfer of the nucleic acid into the host. They act by blocking, as we have said, the M2 channel. It inhibits penetration of RNA viral particle into the cell, into the host cell. That's almost the same thing that we had said. That is the structure of the adamantine. You can see this here coming there down this ring like that. And then there is this link coming like that. And then there is this one. So there is this coming like this. Let's see. I think we can have this coming like this, like that, like that, like that, like that. And then we can have this other one. They have a common, remember, when you have bicyclic rings, they will always have a common center. It can come like that. Those are the ones I'm seeing coming like that and like that and like that. Then there is this one now here, coming all the way down, this way, that, with that. So I can see, as I said earlier, about 3 rings, tricyclic ring. Structure-activity relationship. Amantadine is an adamantine amine. Alpha amino derivative of adamantine is alpha amino is amantadine. N-alkyl and N-dialkyl derivatives of adamantine exhibit antiviral activity similar to that of adamantine hydrochloride. So N-alkyl, so you can actually have here, it's an NH2. So the NH2 can have an alkyl group, or you can have N-dialkyl, where you have the nitrogen having a methyl group and another methyl group. Derivatives of adamantine exhibit antiviral activity similar to that of adamantine hydrochloride, except glycol derivatives and acyl derivatives show decreased antiviral activity. So the only ones that have been shown to have good activity in case you do glycol derivatives. But you can see putting in a glycol moiety, that's a huge moiety. I don't think it has any big advantages over adamantine, so you don't bother. So the other N-acyl derivative show decreased antiviral action, replacement of the amino group with the hydroxyl group, diol group, nitrile group, or halogen produced inactive compound. So that is a very simple structure-activity relationship. So that's why we have said you can have an alkyl group, S4 imantidine, with increase in activity. What are some of the clinical applications? It is effective in preventing and treating all type A strains of influenza, particularly type A2 strain of Asian influenza virus, and to a lesser extent German measles or Latoga virus. It is also used for Parkinson disease. Of course, we did say we do treat the symptoms of course, the problems of the symptoms of Parkinsonism. Generally, at therapeutic levels, it may cause CNS symptoms such as nervousness, confusion, headache, drowsiness, insomnia, depression, and hallucinations. We do understand why, because we did say it releases dopamine, and therefore increases the concentration of dopamine, and the dopamine could be the one responsible for some of these side effects. The GI side effects include nausea, diarrhea, constipation, and anorexia. can also cause conversions, coma, occur with high doses, and in patients with cerebral sclerosis and convulsive disorder. So when we look at it, we are saying most of the side effects, of course, are because of the other mode of action, not this one, which is acting on the virus, where it is blocking the M2 channels and where it has having conformational change and on the hemagglutinin. That is not the main problem for the side effects. The main problem is that because it moves into the vesicles where you have the catecholamines stored, especially dopamine. Releasing that dopamine, there is very high dopamine levels at the synaptic levels, and therefore it will cause the side effects. So that is one class of drugs that mainly affect the uncoating and even early penetration of the virus into the host cell. The other class is the neuraminidase inhibitors, and here we have two neuraminidase inhibitors, and here we have two of them. And you can see now we are having, they are blocking a specific enzyme. This neuraminidase enzyme is specific for viruses, and it is supposed to cleave. When the virus comes, it joins a specific receptor, usually sialic acid, within the host. And now when it joins, it's supposed to have that enzyme which cleaves and allows the virus to get into the host cell. So the influenza virus binds the target cell, the interaction of its surface glycoprotein, which we are calling the hemagglutinin, with the host cell surface receptors that contain sialic acid. So within the host cell, you have sialic acid. Within the virus, you have the hemagglutinin. So the hemagglutinin, a glycoprotein, will bind the sialic acid, usually through, the hemagglutinin has some sugar moieties. So through its sugar moieties, it can bind the sialic acid through even glycosidic bonds. And all we are saying is that this enzyme is very important because it will cleave those bonds and allow. So it is like when this virus comes, it must identify the cell, bind there, and then there is a cleavage, and it is able to penetrate. It's like you form a pole. Once you cleave, you form a pole, you form a channel, and this virus is able to move inside into the host cell. Neuraminidase catalytically cleaves glycosidic bonds between terminal sialic acid residues and adjacent sugars on the hemagglutinin, allowing the virus to leave the cell once the virus has replicated, and even for it to get in. So once it has replicated, and even when it's getting in, that same process is also repeated. So once it has replicated, it also has to get out. That process has to be done. When it's coming in, also the process has to be done. Because the other one, we talked mainly about uncoating. Inhibiting the virus prevents the virus from leaving the cell and infecting others. The hemagglutinin neuraminidase protein, which is responsible for the virus attachment, interacts with the fusion protein in a virus type specific manner to induce efficient membrane fusion. So you can see the hemagglutinin protein, which is responsible for the virus attachment, interact with the fusion. So it comes in. So you have the hemagglutinin, you have the sialic, they are coming in, they are fusing together. And once they fuse together, they will allow this, they will allow for fusion and for the virus to get in. And also when it's getting out, the same process happens. That still, remember, the virus still has its hemagglutinin, the cell still has its sialic acid. So they bind together, cause conformational change, and you're supposed to have pores formed, and the virus exits, is able to exit the host, and of course go and attack other host cells. So once now you block the neuraminidase enzyme, then that means once the virus comes and gets and sticks and attaches itself to the host cell, it cannot move. It will be fixed there. It doesn't have the enzyme to cleave and form the transformational change and move in. And also when it's coming out, when it comes and joins, the neuraminidase enzyme has been blocked. It cannot also have the exocytosis, and therefore it cannot infect other cells. The hemagglutinin of influenza viruses is a major glycoprotein and plays a crucial role in the early stage of virus infection, and of course also after, of course, living, so that they can go and infect other viruses. The hemagglutinin is responsible for binding of the virus, the cell surface receptors, and intermediates liberation of the viral genome into the cytoplasm through membrane fusion. So I hope you can see we are talking about two things. As it enters, it will require its hemagglutinin to fuse or to attach itself to the sialic acid. And of course, we have said that there will be a conformational change, and there will be formation of pores, and there will be endocytosis. And the virus will move in. Once it is going out, the same case, it has its hemagglutinin bind with the sialic of the cell, host cell, again, conformational change and exocytosis, and again, the virus moves out. The key differences between hemagglutinin and neuraminidase is that hemagglutinin binds with cell surface sialic acid on target cells to facilitate viral attachment to host cells. Neuraminidase cleaves the sialic acid from viral receptors to release the progeny virus from the host cell. So one is an enzyme, the other one is a protein. The other one is a protein which comes in, binds the sialic acid. It could bind it through even its sugar moieties, through glycosidic bonds. And once it first of all binds, then the neuraminidase enzyme is the one that cleaves. And once it cleaves, you'll be left with, you can see this is the virus now. This is our hemagglutinin. It has come in and bound our sialic acid. And now our enzyme will come and cleave. And of course, your virus will now be inside, and your sialic acid will still be left intact on the cell membrane. So you can look at it this way. You can see this is our influenza. It has a hemagglutinin. It has a neuraminidase enzyme. Of course, we talked about it has the M2 channels, which are allowing for the uncoating. So once it comes here, you can see this. It is bind. These are sialic acids. So it is binding here. So it is binding the sialic acid. Once it binds the sialic acid, we say there is conformational change, and it allows for like a channel, and the virus is able to move in. Once it is inside, M2 channel inhibitors like amantadine, so there can be now uncoating, so the blocking and coating. And once the uncoating happens, then of course you're free. The uncoating has happened, and you have your nucleic acids. Of course, you have the ribosomes and everything else that now is released into the cell, and that allows now for the other activities to go on. Now, once you have formed, now again it's coming in here, the cell, it binds. So you can see this one was a dent towards the inside. This is a dent towards the outside because it's an exocytosis. This is endocytosis. So now the viruses are ready, and as the virus comes now, again it binds, and after it binds, it again, neuraminidase cleaves, and it's allowed to move out. And the type of the ones we have neuraminidase inhibitors. We have oseltamivir and zanamivir. Those are the ones I have discussed. So you can see this is zanamivir. So what we are saying, let us understand that these ones are binding the neuraminidase enzyme. The importance of the neuraminidase enzyme is to be able to cleave the bond that is formed between the hemagglutinin of the virus, which is a glycoprotein, and the sialic acid of the host. So the sialic acid of the host, and once you cleave, then the virus is able to find a way and move in. And the same case applies when it's moving out. So and of course, the neuraminidase enzyme is specific, as we have said, for the virus, and therefore selective toxicity. So you can see one of them, this is zanamivir, and that's the structure there, and that's the structure of oseltamivir. They say that zanamivir binds the receptor. That is, it binds the receptor, so it will go. So this drug will go for the neuraminidase enzyme, bind it, and therefore prevent it from being able to bind the sialic acid. So they will go, and they say that zanamivir binds more firmly to the neuraminidase enzyme, and there is less likelihood of resistance. Oseltamivir would require conformational change on the receptor or on the receptor, which is a neuraminidase enzyme, and it doesn't bind very firmly, and therefore there is higher likelihood of resistance with oseltamivir. This is just because it doesn't bind as well, does not fit as well into the sialic acid binding site of the viral neuraminidase, and therefore, so it doesn't fit well where the sialic site binds. The sialic binds on the neuraminidase. So these drugs are binding the neuraminidase where the sialic acid is supposed to bind. And once they don't bind well, there is likelihood of resistance as compared to when they bind very, very well. Oseltamivir is fast orally active neuraminidase inhibitor. It's a prodrug compound. There is hydrolysis of the ester takes place in the body to give the active carboxylic acid derivative of oseltamivir. It's a structural analog of a key intermediate in sialic acid chemistry. Oseltamivir serves as a competitive inhibitor of the viral neuraminidase by binding strongly to the active site of neuraminidase. So please remember, it is binding on the neuraminidase, not on the sialic acid. It's binding on the neuraminidase, which is the enzyme that is very important. The viral neuraminidase binds. Once it binds there, then the neuraminidase is not free. It's not available to bind the sialic. If it's not available, then the virus cannot be able to get in, cannot be able to get into the cell or to the host cell. And indeed, if it does bind it when already has been assembly, if it binds when there already is assembly, then again, the virus will not be able to leave the cell. The interaction ultimately prevents the release of the new viral particles from the host cell. Is not a natural product, but it's derived from a natural product called shikimic acid. So there are those two areas where it can bind. It binds the neuraminidase enzyme. Neuraminidase enzyme is not available for cleaving, and therefore this will not penetrate. There will not be fusion. This will also not leave, and therefore the propagation of the viruses will be halted. Maximum binding to neuraminidase occurs when the carbon-6 substituent is substituted with a non-polar chain. If you look at this drug, this is a chain we are saying here. That one there. It is the 3 position. Position this carboxylic group, so we give this first priority, position 1, position 2, position 3. So you can see that this non-polar group is 3-pentyl group. So you can see that position 3 is bound by, when carbon-6, it should be carbon-6. Let's see. The 3-pentyl group 1. This carbon-6 substituted the non-polar chain. Well, if this is 6, that means we are starting counting from here. For us to get this as 6, 1, 2, 3, 4, 5, 6. Sometimes, so it occurs when the carbon-6 substituent is substituted the non-polar chain. So if we are looking at carbon-6 substituent, that means we are counting from here and giving this as carbon-1, carbon-2, carbon-3, carbon-4, carbon-5, carbon-6. Most probably so that we give the double bond. We give the double bond within, so that we give the double bond within the benzene, the cyclohexene ring. We give the double bond the lowest number possible. So we start with 1, 2, 3, 4, 5, 6. And therefore, and you can see we have the pentyl. You look at it, we are calling it 3-pentyl because you say 1, 2, 3, 4, 5. So we are calling it 3-pentyl because if you look at it, that's position 1, position 1, position 2, position 3, position 4, There's another one. 1, 2, 3, 1, 2, 3. And then I think there's another one. 4, position 5, position 6. Why it is important to say it is 3-pentyl, 1, 2, 3. This is 3 here. Why it's important to say it's 3-pentyl? Because you can have 1, 2, 3. It can bind from here. And of course that the binding can give us different positional isomers. So that's why we are saying 3-pentyl, because it can join from there. If it joins from here, that will be the same as joining from here. It can also join from here, 1, 2, 3, and that's why it's necessary for us to indicate which position it's joining, so that we are sure it's joining on that one. So it's 1, 2, 3-pentyl. So it's at position 3 and position 6, and it's a 3-pentyl. So because you can have positional isomers, constitutional positional isomers can be formed, and therefore you need to be specific in saying where exactly at what position the pentyl group joins the cyclohexene ring. So that we are saying that when you have a 3-pentyl group there, then the oseltamivir was found to have maximum activity. Of course, this group is increasing on the lipid solubility, and of course, giving the drug better penetration. If you look at it, it is an ethyl ester. If you look at it here is our ethyl ester here. Again, remember, when you esterify drugs, you block the carboxylic group. You can see this was a carboxylic group here, and you have brought in ethanol. So if you give it as a carboxylic derivative, for one, you can see this will be ionized at physiological pH. It was also an amino group there, which will also be ionized. So it will be as with ion, and therefore very poor penetration of the lipid membranes. So we can't see, therefore there is necessity to derivatize this drug. It has been derivatized into its ester to improve, of course, on its pharmacokinetics, and then, of course, also derivatized into its salt to also balance on the water solubility. So an important feature of oseltamivir is the ethyl ester, which makes the drug orally efficacious. Of course, we have said if you do not do so, it will be protonated, it will be ionized, and once it is ionized, then it will not be able to penetrate the lipid membranes, and it will not have any oral activity. This drug is fast orally active. It's a fast orally active agent for use for influenza A and B. We did say for amantadine and imantadine, they're only for influenza A, both oseltamivir and zanavir all for influenza both A and B. It is also indicated for the treatment of acute illness. If administered within 2 days after the onset of influenza symptoms, the drug is still effective. And also, oseltamivir is actually a prodrug in its ethyl ester form. It undergoes ester hydrolysis to release the active oseltamivir molecule. We are saying even if you give it within 2 days after the onset of influenza, because we have said it will also help stop the viruses from moving from the host cell. So even if you give it just 2 days, as long as the viruses have not started moving out, it will stop them from moving out, and hence it will have very good activity. We are saying it is a prodrug, and we have shown the hydrolysis gets there. Bad water, hydrolysis gets there, and of course you get the oseltamivir carboxylate. Of course, hydrolysis, you do have water, and we have said that helps us in increases the lipid solubility and gives the drug oral activity. It's used to treat symptoms caused by flu and virus. It helps make the symptoms such as stuffy nose, cough, sore throat, fever, chills, aches, tiredness, less severe, and shortness, and shortens the recovery time by 1 to 2 days. Of course, it has its own side effect: nausea, vomiting, diarrhea, headaches, and so on. But most of us, when it is, if it's just the flu, most of us kind of survive. But sometimes flu can be very, very, very challenging. So you are saying you can use that drug oseltamivir to help that. You can use zanamivir. You can use zanamivir. Again, if you look at zanamivir, it has a bit of structural similarity, but you can see it has the guanidino moiety. So zanamivir has the guanidino moiety. And of course, the structures are not really similar, very different structures. Again, the other one was a cyclohexene. The other one was a cyclohexene. You can see this is an oxane. It has an oxygen, and it has an oxine, an oxygen, and of course, it has unsaturation. Of course, it has carboxylic group, just like even the other one did. But this has a guanidine moiety, which was not in the other. So when we look at them, we cannot say that these drugs have much structural resemblance, like the way you are looking at the other drugs. And you look at a whole class, and you can see a pharmacophore, quite a number of these drugs. You don't really, apart of course from the ones we are looking at, the nucleic, the nucleotide reverse transcriptase inhibitors, where you find that they resemble the nucleotides. Quite a number of these other drugs, they have a lot of structural differences. And indeed, that's why we do not have much of structure-activity relationship with them. Effective for both influenza A and B, it has poor bioavailability and poor plasma protein, must be plasma protein binding, and it is used as an oral inhalation. Because of the poor bioavailability, you use it as an oral inhalation. Remember, for the other one, we have said that other one, we can use it. For oseltamivir, we can use it as an oral preparation. But do remember, we say oseltamivir has lesser activity or is more susceptible to resistance than zanamivir. Than zanamivir. It has lesser. Zanamivir binds the receptors, as we said, much more strongly, and oseltamivir giving us much better activity. It is effective when administered before or after exposure. Again, as you have said, the mode of action is the same. It can prevent it coming in. It can also prevent them coming out, and therefore can act at both times. If administered before exposure to the virus, the drug reduced viral propagation, infectivity, and disease symptoms, because it will stop the viruses from infecting the cells. So it will stop viral propagation, also infectivity and disease symptoms. If administered after exposure, the drug reduces propagation, viral titer, and illness. Zanamivir is marketed as a dry powder for oral inhalation. It is used in adolescents and adults. So we are saying the two drugs have very similar mode of actions. They have also similar applications. It's only that we have said also zanamivir has lesser resistance than oseltamivir. Any questions up to there? Questions? So the category of drugs, I think they are just a bit of HIV. I don't know whether we can finish today. No, we can't. So I think, because now we will go to another category where now that one, the ones we were looking at, those that inhibit penetration and RNA replication are for the DNA viruses, the influenza. But now we also look at those that inhibit fusion. Of course, when they inhibit fusion or entry of the HIV viruses, and that's where we look at the docosanol, we look at envavirtide, and we look at maraviroc. So I think because that's another new class of drugs, of course, the same, almost again having an effect on the entry, but now of the HIV virus. The others, as I said, we are looking at influenza A and influenza B. So I think because it's almost a new category, we can look at that tomorrow, because we will continue. We are not far away from finishing. We will continue with the same more. Usernames. Any questions? Can we sign in? I think tomorrow we should still, we are not actually far from finishing. We should finish the entry. We should finish the entry of the fusion inhibitors, look at integrase inhibitors, and I hope look at protease inhibitors. We can do that. There. But protease inhibitors that we should be able to do tomorrow. Then I think we can finish the next lesson we meet, because we only be left with interferon and also looking at the combination therapy. For the combination therapy, you are the ones who will educate me. You are the ones who are doing pharmacology and clinical pharmacy. So you'll be able to tell me which are the current combination therapies, what are the advantages, what are their disadvantages. So please make sure you also look at that, the current combination therapies. We are not looking at in other countries. We want to understand Kenya. So what are the current combination therapies? Which drug come in combination? How do we combine them and why? And what are the conditions that govern which combinations will be given to which clients? What are the what you look at? What is important for you to decide on which combination to give to which patient? Because there must be factors that are very important in making one decide. And that is why you will find that some of the drugs are in certain combinations, but you get all the individual drugs on their own, because there can be very difficult patients whereby you cannot give certain combinations, but you can give them specific combination because of other underlying conditions or other drugs they are having, or because of the fact that they have had a lot of, when they are exposed to some of the drugs, let's say like the protease inhibitors, they have a lot of toxicity. So you individualize the drugs for them to see whether you can minimize on the toxicity, increase on the effectiveness, and of course also consider the cost.